You can use that now.
Okay, thank you.
Sometimes once you forget it, it will actually work.
Okay. Maybe I just wasn't clicking hard enough. So I don't have any disclosures or conflicts of interest other than the fact that I do run a blog about gut health and I have a Patreon account to help support that. We all know here that the gut microbiota is implicated in chronic disease. In many cases, this is just correlational or association studies. The gut microbiota seems to be altered in these various different chronic diseases, but in many cases, we've also got fecal transplant studies now into germ-free mice that have shown that some of these relationships are in fact causal, or at least partly causal. Some researchers have hypothesized that the loss of ancestral diversity is actually what's driving these chronic diseases. So some of you may have heard of Dr. Martin Blaser, who wrote the book Missing Microbes. He's done a lot of research on this. And essentially his theory suggests that the loss of particular ancestral microbes may impair the normal metabolic, cognitive, and immunological development of the host. So, and these changes are cumulative across generations and associated with increasing disease risk. So here on the right, you can see this, oh dear.
You pressed the wrong one.
I definitely pressed the wrong one. There we go. Okay, sorry about that. So here on the right, you can see that there's this progressive loss of diversity with increasing westernization, with the US being here on the lowest line here.
So what's your axis?
So this is bacterial diversity, and this is essentially an alpha rarefaction curve. So we're mostly looking at these final time points here to assess total diversity, since the diversity will essentially, the number of species you identify will plateau as you sample more of as you sequence more of the sample. So here you can see this is the Yanomami tribe, the Gushibo, Malawi, and then USA. And so this was with increasing westernization, we see this progressive loss of ancestral diversity in the microbiome. So by some estimates such as this, we've lost about 50% of our diversity. And of course, there's a number of reasons for this. Perhaps the most obvious and well-known, perhaps, is the overuse of antibiotics. So these were no doubt one of the greatest discoveries of the 20th century, but they are widely overused, devastate gut microbial communities, and recovery is often incomplete. Still struggling with this button. Recent studies have also suggested that non-antibiotic medications can also alter the gut microbiome. In fact, about 24% have a noticeable impact on gut microbial communities. How detrimental those changes are is not well characterized yet, but at least 24% are having a significant impact on the community composition.
Could you give like any particular examples from that 24%? What the common ones are, or how they do it?
Yeah, surprisingly, it was kind of all across different categories of pharmaceuticals. So it wasn't really like, we would expect things like PPIs to have an effect, things like immunosuppressants to have an effect, but it was much more widespread than that. Antidepressants, antihypertensives, pretty much within each category of drug, there were a number that did modify the gut microbiota. And you can check out this paper here for more details on that. I don't know what I'm supposed to be pointing at. Maybe that's the trouble. Another reason potentially for loss of diversity is the loss of seasonality in our eating patterns. So this was a really interesting paper published in 2017 in the journal Science on the microbiome of Hadza hunter-gatherers. And they found that they tracked their microbiota over the course of a year and a half, and they found that the composition of the microbiota and its function reflected the seasonal availability of food and what they were eating. So there were striking differences between the wet and the dry seasons, with many taxa dropping to undetectable levels during one season, only to flourish in the next season when the substrates that they were eating changed. And what's interesting is the same microbes that were most likely to oscillate between the two seasons in the Hadza are completely absent in our industrialized populations. So there's a number of other things I could talk about that are potentially leading to our loss of ancestral diversity, lack of time outside, just disconnection with nature, not being in the soil. But I'm kind of going to focus more on how we can improve our gut ecosystem. And I think it's really important that we look at the gut as an ecosystem. So a few years ago, I went to a talk by Dr. David Wellman from Stanford, who uses this analogy of your gut as a rainforest. And so essentially, this is your healthy gut, and then this is your gut on antibiotics. And so there's a couple key points here. The first being the environment is really what determines which species can survive. And this is why we have a completely different microbiome on our skin than we do in our guts or any other region of the body. It's because the environment and what's present for the microbes as substrates really determines which species can survive. I'll come back to that idea a little bit more later. We also can't just look at one or two species to determine the health of the ecosystem. Today, there's a lot of people getting excited about one bacteria or another and looking at their abundance on microbiome tests and saying, oh, I have an unhealthy microbiome, I have loads of phytobacterium. They're not really looking at the whole of the ecosystem. So it's really important to consider how microbes are interacting with each other and with the environment. And another thing I want to stress is that there's no one healthy microbiome. There's multiple stable states, multiple healthy microbiomes. And just like there's not one healthy rainforest, right? So there's going to be differences. Genetics, geography, diet are all going to have an impact on your unique microbiome. We actually only share about 1/3 of our microbes with our neighbors. So another way to think about this is what's called a community stability landscape. And so in this circle here represents the gut ecosystem, right? And essentially this valley it's sitting in represents a stable state. So you can imagine that if it gets to a peak, it's going to fall into another valley, which is another stable state. So a perturbation like antibiotics could potentially get you out of this stable state by overcoming this resilience you have to disturbance and potentially drop you into a disease state. Is that making sense to everybody? So once we're in the disease state, the question is, how do we get back to that healthy state? And we kind of have to overcome that threshold to be able to get back into the healthy state. So of course we can think about lifestyle factors, our ancestral health principles. And for a good chunk of the population, if we do diet, exercise, sleep, circadian rhythms, perhaps a little bit of fasting, we can essentially perturb the system back into that healthy state. Maybe not the exact healthy state it was in before, but a much healthier state. And we do know that these things have a major impact on the gut microbiota. So dietary changes are known to rapidly shift the gut microbiota in less than 48 hours. This was a study done by David et al. Published in Nature, where they shifted people from a plant-based diet to an animal-based diet and then back again, and they showed rapid changes in the gut microbiota composition. Here they're showing beta diversity. But in terms of a number of other measures, there were significant changes in terms of the bacteria that flourished and the bacteria that recessed into lower abundance. There were also alterations in microbial gene expression, which is not too surprising when we see such drastic changes in the composition. And of course, Bob covered this in his talk a little bit, but dietary fiber is fermented into short-chain fatty acids, butyrate, acetate, and propionate being the most prevalent in most human guts. I'm going to focus mostly on butyrate for this talk, because butyrate helps maintain gut barrier function. So it increases mucus secretion, epithelial proliferation, and turnover, promotes release of antimicrobial peptides, which help to regulate the composition of the gut microbiota, and also maintains physiologic hypoxia and gut homeostasis. And I'm going to come back to that idea a lot more later in the talk. I also want to point out, I guess Paul's not here, but any other carnivores in the audience, before you start throwing things at me, ketones and isobutyrate can also make up for low butyrate. Isobutyrate is a metabolite of protein fermentation, and it can stimulate a lot of the same receptors as butyrate in the gut to exert those anti-inflammatory effects, those mucus protective effects. And importantly, acetoacetate and beta-hydroxybutyrate, those ketone bodies produced during a high-fat, low-carbohydrate diet, can also potentially supplement these colonocyte energy pathways. And a lot of people don't necessarily know this. So here we can see, this is kind of the typical pathway that we know is dietary fiber. We have those microbes that ferment dietary fiber into butyrate. Butyrate is then uptaken into the epithelial cell and then goes through its metabolic pathway down to producing energy for replication. Of course, it also has signaling functions, which I mentioned, but this is the main way that butyrate provides 70% of the energy for colonic epithelial cells in the healthy gut in people who are primarily eating carbohydrates and fiber. But in the case of gut inflammation, we actually might have either we don't produce enough butyrate because we don't have the butyrate producing bacterial populations to support the production of enough butyrate, or we have inflammation of the gut mucosa, in which case butyrate uptake might be reduced, as has been shown in inflammatory bowel disease, in which case it might be better or ideal to be able to get some ketones into this pathway because they actually come right in. Butyrate is actually converted into essentially beta-hydroxybutyrate and acetoacetate before it's used for energy through beta oxidation. So ketones can potentially make up for low butyrate, and we'll come back to that idea as well. Research that I did as part of my graduate dissertation work also shows that exercise has independent effects on the composition and function of the gut microbiota. So in animal models, we've shown that exercise increases microbial diversity and butyrate producers, generally increases short-chain fatty acid production, and increases some of these bacteria we think to be beneficial. Cross-sectional studies have also looked at athletes versus non-athletes and looking at differences in diversity and composition there. And we see an increased abundance of a number of key species that we associate with health, Faecalibacterium prusinense, one of the keystone butyrate producers in the gut, and Akkermansia muciniphila being a protective mucus dwelling microbe. But of course, we didn't know from that whether that's because athletes eat healthier or if it's a real effect of exercise. So in 2017, we did the first longitudinal study of exercise training on the gut microbiota, and we found that especially in lean individuals, it did increase fecal butyrate and the abundance of butyrate producers. If you're interested more in the exercise microbiome connection, I have a number of blog articles on it, and I also wrote a review paper that covers all of these studies and a few more.
Can you commercially get a level of fecal butyrate?
Sorry?
Fecal butyrate, can you get that in patients or anything?
Can you test it, you mean?
Yeah.
So you can test fecal butyrate. It is unfortunately the best proxy measure we have for gut butyrate, but it's not perfect because it's basically a measure of how much butyrate you're excreting. So it doesn't tell you how much you're producing or absorbing. So it's a function of all of those things, which is why it's helpful to have animal models as well, where we can more directly measure butyrate production. But you can measure fecal butyrate. It is a complicated methodologically. It's complicated, so a lot of stool testing companies are doing it in a way that's probably not preserving the butyrate enough until it's measured. So there are some issues with measuring that clinically, but could certainly be one indicator in determining gut health. So I want to go back to this community stability landscape because so if these diet and lifestyle strategies aren't enough, we might add some other interventions in. So this is kind of what we think of in functional integrated medicine, add in herbals, maybe we try some probiotics, prebiotics, or even go as drastic as FMT. But the question is, well, for another chunk of people, this is going to move them back into that healthy state. But the question is, what happens for the people where this doesn't occur? Quick note, if you're interested in more on those more basic strategies, I did cover them in my AHS talk, which you can find at this link here, and I'll be sure to share that after as well. But I really want to focus on what happens to people who can't get over that hump with those strategies. Because we all know people like this, people who seem to have tried all those strategies and often have been working on their gut health for years, seeing practitioner after practitioner, and still stuck in a state of perpetual dysbiosis. And often they've done multiple rounds of antibiotics, herbals, FMTs even, and are still having symptoms. This actually describes about 10% of the clients I work with. So my major research questions, thinking of that, is how can we reverse severe gut dysbiosis in an individual once it has occurred? And how can we increase resiliency or prevent dysbiosis when faced with a disturbance? Once we're in that healthy state, how can we keep ourselves in that healthy state? And so to answer these questions, I want to first kind of cover what dysbiosis is and why it occurs. So gut dysbiosis is a pretty loosely defined term that's thrown around, but it's generally refers to an altered state of the gut microbiota, usually associated with disease. And since everyone has a unique microbiome, there are virtually infinite states of gut dysbiosis. But there are some signatures that seem to be common across different diseases. And so this review is really excellent by Litvak et al. They say perhaps the most consistent and robust ecological pattern observed during gut dysbiosis is an expansion of facultative anaerobic bacteria belonging to the phylum Proteobacteria. Let me explain this a little bit more in a second. But essentially here you can see that there's, this is a healthy gut homeostasis state where you have primarily obligate anaerobes. We would probably have about 1 to 3% proteobacteria in there that's not shown, but predominantly obligate anaerobes, whereas here we have an expansion of proteobacteria, those facultative anaerobes. We often see with that high proteobacteria, there's often a concomitant reduction in the butyrate producers. And again, we see this across a number of different diseases. These are just some that this pattern has been found. And again, so the healthy microbiome is predominantly those obligate anaerobes. And to remind anyone who's maybe not familiar with this, these are microbes that can only grow and reproduce in an environment that is devoid of oxygen, or very, very low in oxygen. It includes many beneficial bacteria, including those that produce the short-chain fatty acids like butyrate from dietary fiber. Then, of course, we have the facultative anaerobes, and these are capable of growing in an environment with or without oxygen. It includes many opportunistic pathogens, mostly in the phylum Proteobacteria. You may recognize a number of the ones listed here. So these are present in the normal healthy human gut at low abundance. But the problem becomes when they expand, as we'll see more going forward. So a really fascinating body of research to come out of the labs of Dr. Andreas Baumler and Dr. Sebastian Winter that I came across recently suggests that colonocyte metabolism and oxygen leakage into the gut is what is actually driving the expansion of these facultative anaerobic bacteria and essentially causing dysbiosis. So it really starts with a depletion of epithelial energy substrates, which then results in oxygenation of the gut mucosa. And this results in a downregulation of a gene called PPAR gamma and a shift towards an inflammatory colonocyte profile. So if you look at this figure on the right here, so this is a healthy colonocyte. We have fiber being fermented by the microbiota into butyrate. Butyrate is then uptaken into the epithelial cell, and it's activating this gene, PPAR gamma, which is kind of the control switch of metabolism. And when we have activated PPAR gamma, it's stimulating fatty acid oxidation through beta oxidation in the mitochondria, and beta oxidation of fatty acids like butyrate, actually. So it's stimulating basically its own oxidation, uses high amounts of oxygen. So we have oxygen diffusing in from the blood to the epithelium. And normally that oxygen is primarily used up in the cell through beta oxidation. And this keeps the epithelial cells pretty happy and in a state of no inflammation. But essentially what happens when we either don't have that butyrate substrate, or when we have some kind of other inflammatory agent here that's causing inflammation, essentially what happens is there's no butyrate being uptaken, no activation of PPAR gamma. We're then getting oxygen here that should be being used for beta oxidation, but instead we're doing anaerobic glycolysis. So instead glucose, since there's no butyrate, essentially the epithelial cell is then starving. So it pulls glucose from the blood instead and does anaerobic glycolysis to lactate fermentation. And then it's not using the oxygen that it should be for beta oxidation. So then that oxygen and lactate ends up getting very concentrated in the cell, and it ends up kind of spewing out into the gut lumen. And this is where oxygen and lactate can really feed pathogenic bacteria. I know I'm getting a little in the weeds here, and I'm at the risk of being in quadrant 3 and 4. But I think it's really important to understand why dysbiosis is occurring, especially for those people who the quadrant 1 interventions, regardless of how much they do them, they're really not seeing any improvement. So there's a number of things that can actually cause this shift from the healthy colonocyte to this inflammatory profile. And I'm going to talk about each of them a little bit and highlight some of the studies that have shown this. But it occurs with antibiotic exposure, where you deplete gut butyrate, pathogen invasion, where you're causing significant mucosal inflammation, and potentially I'm theorizing a low-fiber diet in the absence of ketones as well. So the first is antibiotics, and essentially these deplete gut butyrate, so they're leading to a loss of gut hypoxia. So in a normal, healthy state, butyrate enters the epithelial cell, and in addition to stimulating that PPAR gamma pathway, it's also increasing this hypoxia-inducible factor, which is kind of like an oxygen sensor in the cell. And when that oxygen sensor is activated to say we have low oxygen, it coordinates gut barrier protection. But when you knock out the microbes that produce butyrate with broad-spectrum antibiotics, it reduces butyrate levels by about 4-fold, and you get a rapid oxygenation of the mucosa, loss of hypoxia, and impairment of gut barrier function. This was not just due to lack of substrate, so they tried putting fiber in there, but the microbiota, just after antibiotics, had lost its ability to produce short-chain fatty acids from fiber. Interestingly, some pathogens can actually hack our gut metabolism, hack this pathway to promote gut dysbiosis, so that they themselves can take over the niche in the gut. So this is research done with Salmonella typhimurium, which is a common cause of food poisoning. It can invade the host mucosa and cause inflammation. And when it does this, again, we get that host-derived oxygen and lactate leaking out into the gut lumen, selectively feeding it the substrates it actually wants to survive. So Salmonella is causing inflammation and then basically causing the host to leak the substrates that it thrives on. It also tends to, at the same time, deplete butyrate-producing Clostridia, both through direct competition and because you're essentially not getting that butyrate feedback loop.
A second ago, when you said the one before that, I just
Yeah.
When you said that the microbiota lost its ability to produce, is there an indication if that's like because certain strains were lost, or if just like ones that normally could produce short-chain fatty acids no longer?
Oh, it's definitely a loss of those species. Yeah, so they've looked in the mouse model. Clostridia is the main butyrate-producing groups that were knocked out by these antibiotics, and that's what led to the reduction in butyrate. Yeah. So the microbiota composition essentially shifts when the antibiotics hit, and there's no longer those butyrate-producing microbes present. They will, if you stop the antibiotics, they will recover over time, and then the gut microbiota would be able to produce short-chain fatty acids again. But in the, acutely right after the antibiotics, those species are ablated.
Right, so it's the distribution that changes, not the
Right.
individual species' capacity to do these
Exactly.
things.
Great. So far we've seen 2 examples where butyrate depletion led to gut oxygenation and dysbiosis. So given that the #1 source of butyrate is from dietary fiber, unless we're in ketosis perhaps, I think it also follows that it's likely that low fiber intake out of the ketosis context would also lead to gut dysbiosis by the same mechanism. This remains to be shown in a controlled study, but there are a number of studies that have looked at what a Western diet essentially does to the gut microbiota, and it tends to increase proteobacteria. So potentially one of the things that I hope to study in the future is this particular one. So what's interesting is that all of these kind of have in common inflammation, right? Interestingly, a study in 2007 showed that gut inflammation itself is actually enough to disrupt the gut microbiota and promote the growth of proteobacteria. So there's also a number of sources of inflammation that have been shown to increase proteobacteria in other studies, such as dietary emulsifiers, artificial sweeteners, psychosocial stress. And there's also potentially other mediators of gut inflammation. So these are the ones that have been shown in have been published in studies, but we can certainly imagine that other things that would inflame the gut, such as perhaps undiagnosed food sensitivities, lactose intolerance, could also potentially lead to inflammation and promote gut dysbiosis through this mechanism by impairing colonocyte metabolism.
You see, patients often ask me whether stevia is a safe or artificial sweetener. Any data?
I don't think I've seen any on stevia and gut health in particular. I know that there's a number of other studies on other artificial sweeteners that impair glucose tolerance because of how they affect the gut microbiota. So I'm kind of hesitant with stevia, but I haven't seen any evidence one way or the other.
Certainly, if any that's by an insulin response, I have patients who either do or don't get hypoglycemic from stevia. The ones that do, I feel, are eliciting an insulin response and eventually glucose intolerance.
Interesting.
Just taste it.
And then
Oh, yeah, but we get these questions.
Lucy, are there emulsifiers that are more like carrageenan versus guardian? Is there any like a hierarchy of that, or that there's all that?
I mean, so I can't remember which particular one.
I think it's polysorbate 80.
I think it's polysorbate 80
and
for this one.
hydroxymethyl cellulose were the 2 that they studied.
Yeah.
And you'll see something like modified cellulose or something like that on the package insert, and that's what it is.
Yeah. But there's also been studies on sunflower lecithin and things on negatively impacting gut health. Great. So this is kind of the summary of those last few slides. So essentially what we have is kind of we're stuck in this vicious cycle of gut inflammation leading to a loss of gut hypoxia, then feeding those facultative anaerobes, promoting gut dysbiosis, which then, those proteobacteria are then inflammatory themselves and lead to more gut inflammation. So in this vicious cycle, caused by a number of things, antibiotics, pathogenic infection, low fiber diet, or other inflammatory agents.
we can interrupt you for a moment.
Yeah.
Through this thing. Okay, one of the things, I was doing a presentation on the gut microbiome and the risk, as you mentioned. One of the things that I saw, and I think it was in Perlmutter's work, is that if a young person has antibiotics, they return to their natural diversity so much more quickly and reliably than an older person does. And do you think it's just accumulation of all these things, or is there a particular aspect of an age makes it vulnerable.
Yeah, that's really interesting. I would think that it's probably that an older gut is a little bit more susceptible to being is a little less resilient. So if you think about that community stability landscape, that that older gut is going to be
Just for
less resilient to disturbance.
Less exercise.
Right. So studies have shown that the gut microbiota does become more inflammatory with aging, and that the gut barrier function does decline. Now, whether that's a normal function of aging, I don't, kind of relevant to what we were talking about yesterday, or whether that's more pathological aging. That definitely seems to be what occurs. And if your gut barrier function is not already optimal, then something that triggers inflammation is probably going to have a more dramatic effect than it would in a young, healthy person. Great. So I want to get back to those research questions. How can we reverse gut dysbiosis in an individual once it has occurred, once they're in that vicious cycle? And fortunately, we do have some keys from the studies that I presented. So I just want to kind of go back to PPAR gamma and remind everyone that this is really the control switch for colonocyte metabolism. So I showed before those two different colonocytes, the healthy and the disease, but we can also kind of think of this as a spectrum of, epithelial energy starvation. Over here, where we're not getting enough butyrate, PPAR gamma activation is reduced, lower beta oxidation, and reduced hypoxia. So you're going to have more oxygen leaking into the gut, dysbiosis, and inflammation. Over here, if you have plenty of fuel, energy substrates for the gut, butyrate, ketones, etc., you're going to upregulate that PPAR gamma, increase beta oxidation of fatty acids, and hypoxia, leading to gut homeostasis. So the question is, could actually stimulating this pathway prevent or reverse gut dysbiosis? So is there any evidence for that? And in fact, there's quite a bit. In inflammatory bowel disease, rosglitazone is a PPAR gamma agonist. It's been shown to prevent dysbiosis and reduce colitis symptoms in animal models. It's a pretty gnarly PPAR gamma agonist, so it does have a lot of other side effects, and is typically not used much in humans, especially for inflammatory bowel disease. So now the first line drug treatment is mesalamine, which has a little bit more of a targeted effect in the gut, and the anti-inflammatory effects have been shown to actually be mediated through PPAR gamma. And people who go on mesalamine end up with a reduction in proteobacteria and increased abundance of Faecalibacterium, which is that keystone butyrate producer. Interestingly, butyrate supplementation itself for 2 months was also shown to increase abundance of butyrate-producing bacteria in a randomized control trial of IBD patients. So it's interesting because we can actually almost think of butyrate as a prebiotic, because it's, indirectly it is leading to increased abundance of butyrate-producing bacteria.
You believe that somehow is, we go back to quadrant 3-4 slide.
Yeah.
It somehow decreases oxygen into the gut. I believe that's the pathway.
Yeah, I think so. Essentially, if you're providing extra substrate, right? So in inflammatory bowel disease, you're essentially having low-grade energy starvation, right? You're having, basically, you have reduced PPAR gamma expression in inflammatory bowel disease, so you're increasing, the inflammatory colonocyte pathway. So if we provide supplemental butyrate, it can essentially shift back towards that healthy state. Higher PPAR gamma activation, you're getting beta oxidation of that butyrate. You're promoting the healthy environment.
Rosy glitazone? Is that, there's rosy glut of the TZD, the Avandia?
I'm
Yeah, that's what it is.
Okay.
okay. Thank you.
She used that a lot years ago. Okay.
I've only read it in papers and never said it aloud before, so.
It's great if you don't mind getting half
Like,
Right, rosy glitazone was came out first.
Yes,
Yeah.
India.
And this hasn't just been shown in inflammatory bowel disease. There's also in models of diet-induced obesity. They've interestingly found that there's this synthetic version of 2 compounds that are found in a traditional Chinese medicine formula. I'm going to say DBZ, so I don't have to pronounce that. It activates PPAR gamma to a lesser extent than rosiglitazone, and is still able to protect against gut dysbiosis, barrier dysfunction, and body weight gain in that mouse model.
And our drug company can sell
Yeah.
So the DBZ is a synthetic version of the medicine. Is that what you're saying?
Correct. So yeah, so they took 2 compounds that are found in this Chinese
Do you know
medicine
the
formula.
2 compounds?
Yeah, it's a borneol ester, and I think danshenzu is one of them, and bingpian is the other. So the Chinese medicine formula is called Fu Fang Danshen, I believe. So yeah, I honestly, I'm curious why they didn't just use the formula itself. It may be so that they can market it better. But I'm excited to see more studies on how herbal compounds could potentially activate this pathway and be particularly helpful. So actually, I kind of did a review of the literature and looked for any strategies that could potentially target this pathway and support gut hypoxia. And some of these are in that quadrant 1. So we've got, diet, exercise, fasting, and caloric restriction. I guess, oops, I'm button again. Like I said, supplementing with butyrate. I touched on mesalamine and the DBZ compound. Stress management is also important. Stress has been shown to reduce that PPAR gamma activation. And then a number of these herbal compounds and nutrients, probiotics and prebiotics, could potentially target this pathway and support gut hypoxia. Again, this is kind of really focusing on those people that those basic diet lifestyle strategies aren't working. Maybe we could use some of these strategies to kind of tip them back into that healthy state, and then be able to go back to maintaining that healthy state and increasing resilience with diet, exercise, stress management, etc.
I've seen one study of triglycerides or inulin in healthy people. They actually got worse blood glucose control with the prebiotic.
Interesting.
I'm wondering if you have any thoughts on where those isolated prebiotic fibers are useful or not.
Yeah, I generally don't use them. I think, especially in people with inflamed guts, it just tends to increase inflammation. It's just not something we would ever find, right? Is an isolated fiber in its concentrated form. Some people do try them and they tend to see benefits from them. But I typically shy away from doing isolated fiber.
I mean, the same thing could, instead of saccharides, these kind of something too.
Of what? Sorry?
The FOS, your fructooligosaccharides, right?
Yeah.
That as an isolated.
Oh, definitely.
Similar problem.
Where does aloe vera fit in this list? Good bacteria feeder.
I haven't seen anything I didn't find anything about aloe vera and this particular pathway. That doesn't mean it's not helpful to the gut. But this is kind of focusing on things that can really shift back towards that gut hypoxia and stop feeding facultative anaerobes.
No studies on specific foods.
Sorry?
No studies on specific
No
foods,
studies on specific
like
foods.
bilberries.
No, but maybe I'll do them as part of my postdoc. So
If I go to
hopefully.
your blog from that reference there, can I find a study of gas?
Yeah, definitely. So in terms of with Iberogast, it wasn't a study that actually looked at Iberogast itself, but a lot of the components of Iberogast, the herbs that are in it, are known to activate PPAR gamma, and actually, surprisingly, almost all of them do.
Oh, wow.
So I don't know if that was at all relevant in how they chose to formulate that, but Iberogast has been clinically shown to improve symptoms and functional bowel disorders. So it was really interesting that it has so many herbs that do affect this pathway. So I'm really interested in exploring some of these in my future research.
Is there a reason why pyroluism wasn't looked at also? Because that's still on the market. Rosy.
Interesting.
So it's still available. It's
Interesting. I haven't seen anything with that one. I think it has been used in some of the animal models to show that PPAR gamma was implicated in the pathways, but I haven't seen any human studies on how it affects
This is really interesting
gut
because
metabolism.
those were huge diabetes drugs.
Yeah.
Years ago, I used tons of those, and I never really noticed any influence on the gut, but maybe we were just one acid.
Yeah, interesting. Yeah.
Next slide. Given constraints of water environment, if you don't have any gut problems, everything on that
Yeah, that's interesting. I mean, I think periodic fasting and exercise are probably the lowest hanging fruit, right? Going to have the biggest impact on this without potential side effects. So I would never recommend that anyone who's got a healthy gut, go be taking mesalamine to try and make their gut more resilient. I don't, it's still a pharmaceutical, has side effects. I will talk a little bit more about, if you think that if you're going to be taking antibiotics, actually doing some of these might be really beneficial to prevent antibiotic-induced dysbiosis. So I'll kind of get to that in a minute. So I also wanted to make a quick note about the importance of mitochondrial health in this, because even if you've got all the substrates, if you don't have healthy mitochondria to do the beta oxidation, you're going to be shifting to that inflammatory profile. So it's really important that we support mitochondrial health, and mitochondrial pathology has been found in a lot of different gut disorders, especially inflammatory bowel disease. They're essential to that butyrate metabolism to maintain gut hypoxia. PPAR gamma activation itself actually supports the formation of new mitochondria and kind of supports their function as well. But it's possible that if someone is struggling with their gut health, then supporting mitochondrial function with some of these additions might be helpful. Obviously, this is, again, after doing all the diet and lifestyle things to optimize mitochondrial health. So I'm really interested in how we can kind of combine some of these things to create integrative, synergistic approaches to break that cycle and create essentially enough of a disturbance to shift back to that healthy gut ecosystem. So interestingly, some studies have found that mesalamine combined with curcumin or butyrate is more effective than mesalamine alone for IBD. So this is one example where they've shown synergy. Not a lot of other studies are looking at synergy of these different components, but I'm really interested in how we could try integrative approaches. So for example, we might imagine using mesalamine, curcumin, some other things to activate PPAR gamma. We could provide butyrate and ketones to support epithelial energy, and then perhaps something like L-carnitine to make sure that those energy substrates are actually getting into the mitochondria to be metabolized. So this is kind of like a kitchen sink approach, but when you have patients that have really tried everything, perhaps this is something that could actually move the needle and provide enough to shift back. Yeah.
They looked at like exogenous ketones. Is that your
I would love to look at that, and that's actually I'm hoping that I can especially look at ketones and how they affect this pathway. Actually, there are so few studies on how ketones affect gut barrier function. Like, it makes complete logical sense that ketones can feed in and provide epithelial energy substrate, feed into the butyrate pathway, and promote epithelial proliferation. But there's very few studies to show that ketones can actually support gut health.
We can get D'Agostino or Rare Stops to send you the, I mean, they give you
The
stuff
ketone
to
esters,
try.
yeah, definitely.
MCT oil be cheaper?
Sorry?
MCT oil be cheaper?
Perhaps, but maybe not as well tolerated by someone who's already got the gut issues. So it just depends on how tolerated. So I'm currently trialing some of these protocols with my clients. They're working very closely with their gastroenterologists, since there are some of these more potent PPAR gamma agonists being used here. So hopefully I'll have more to report back in a year once I've been able to look into this even more and try it out with people.
So you give them the solutions that actually work, and the gastroenterologist prescribe the PPIs.
Take all the credit.
Well, they'll prescribe them a salamine.
Right,
Yeah,
they prescribe them salamine.
They've done a little bit.
Everybody gets a PPI, I guess.
So I also, what about dysbiosis of the small intestine? So far I've mainly been talking about the colon and the metabolic switch that happens in the colon. But does this actually happen in the small intestine? And this is important because we know that small intestinal dysbiosis, rather than SIBO, actually underlies a great deal of gut symptoms. So for anyone who's not familiar, this paper came out, I think it was last summer, that suggests that a lot of our SIBO knowledge is really misguided, and that more often than not, it's not bacterial overgrowth, it's actually just dysbiosis. So a shift, and often increased proteobacteria, interestingly, which is causing a lot of the gut symptoms we think are associated with SIBO, like bloating, abdominal pain, etc.
I mean, oftentimes locally, clinically, we're like using it to mean the same thing.
Oh, definitely. But in the research world, unfortunately, there's been a real focus on the overgrowth, and a lot of the studies have been defined based on the diagnosis of the number of CFU per mil in cultured aspirates, which is just completely outdated. Because what grows really well in culture? Proteobacteria.
Yeah.
So really what they've been seeing is people who have high proteobacteria, they don't necessarily have more bacteria in their small intestine, but they have more proteobacteria. They're going to be diagnosed with SIBO because that proteobacteria is going to grow really well in the culture, and they're going to think that they have an overgrowth because there's a higher CFU per mil count than they would otherwise. So I agree with you. A lot of practitioners are using them synonymously, but at least in the research world, there's been a lot of confusion over the fact that it's overgrowth, and also a lot of people thinking that because it's overgrowth, they need to kill the overgrowth as opposed to try to modulate the ecosystem and shift it back to healthy. Can this actually affect the small intestine? There's definitely lower PPAR gamma expression in general in the small intestine than the large intestine, and a slightly higher oxygen concentration as well as you move up. But I was able to find one study was actually published in PNAS where they gave they call it a high-fat diet. Of course, don't give any details there, but that's whenever you see a high-fat diet in a mouse study, you can pretty much assume it's a Western high-fat, high-sugar, low-fiber diet. Yes. Yeah. So they did find that this Western diet downregulated PPAR gamma in the small intestine almost twofold, leading to reduced antimicrobial gene expression, which is what regulates the gut microbiota, especially the small intestine, and caused gut dysbiosis of the small intestine, which was reversed by rosiglitazone. So that was really interesting, and I'm really, really interested to think more about how this could be implicating in the small intestine. So thinking about that a little bit more, butyrate is the major fuel for epithelial cells in the large intestine, but glutamine is the primary fuel in the small intestine, and it activates PPAR gamma in the small intestine, much like butyrate does in the colon. Could we provide glutamine to people who are having the small intestinal dysbiosis and potentially help overcome that epithelial energy starvation? There's also a question of whether we could use certain mesalamine formulations for IBS. Most studies have found fairly low efficacy. It's been kind of mixed. But those were mostly using lower doses. Recent study using a higher dose, which is closer to what's used in inflammatory bowel disease, for 12 weeks had significant benefit in IBS-D patients. And I also wonder, it's definitely going to be individual because IBS is a very heterogeneous condition, but it's possible that, you know, going back to that integrative approach, synergy, maybe we could combine mesalamine or that DBZ compound with glutamine and ketones to really provide that support for the small intestine. So that's really going back to, as opposed to trying to modulate the gut ecosystem or kill bacterial overgrowth, perhaps we just need to support the environment, you know, from that ecological perspective, support the environment, and it will select for the microbes that we want.
So I'm sorry, you had ketones on there also, but you're saying do the same thing as substitute as glutamine or for the small intestine as well?
Correct, correct. So I believe I found one study that suggests that ketones can feed the small intestinal epithelial cells. Whether it can increase PPAR gamma there,
right,
I'm not sure. Ketones generally increase PPAR gamma across multiple body tissues. So some of this is more speculative, especially for the small intestine. But it really doesn't make sense, you know, if we went through periods of fasting, that our cells wouldn't be able to use ketones in the small intestine as well. So I'm going to summarize that part there, and then I have just a little bit more on resilience. So the first is we need to think of the gut as an ecosystem, importance of the environment in determining species abundance, rather than directly just trying to modulate the gut environment. And I think we have been doing this to a great extent in terms of trying to do things that support gut barrier function, but perhaps we need to be thinking about the hypoxia piece as well. High proteobacteria, low butyrate producers is a signature of gut dysbiosis, and when you see this, it likely indicates that you've got that underlying epithelial metabolic dysfunction and inflammation. Antibiotics, gut infections, or low fiber intake without ketones can all deplete gut butyrate and lead to this vicious cycle. This understanding of how oxygen drives gut dysbiosis offers important insight on how we might be able to reverse it, and I'm hoping that I'll be able to do my postdoc research in this area as well. If basic gut health interventions fail, targeting PPAR gamma pathway may be able to help us get back to that healthy state using integrative synergistic approaches. And then, of course, we can hopefully still maintaining those ancestral health principles and being able to use those to maintain and promote health and resilience. So I'm going to briefly touch on the second research question, which is how can we increase resiliency or prevent dysbiosis when faced with a disturbance? So of course, antibiotics are perhaps the most significant disturbance that we know today. Sometimes unavoidable. Sometimes you get bit by a snake in Costa Rica, right?
They were all my bees, I think. Like all my antibiotics were.
Oh, interesting.
Yeah.
Probably still has an effect on the gut.
Antibiotic, whether it goes across the gut epithelium, it's not antibiotic, it's the antibiotic dependent.
You're better
Perhaps.
off. Good job.
Perhaps.
I guess I wasn't in a position to use the antibiotic. So
No, definitely not. Definitely not. I mean, yeah, so sometimes antibiotics are unavoidable, but they can cause a loss of diversity and potentially increase risk of chronic disease, especially if we get them during early life, where it's a critical period for the maturation of the gut microbiota. And antibiotic exposure has been shown to increase risk of asthma, obesity, autoimmune disease, skin conditions. I could probably list a whole bunch more here as well. So the question is, what do we do if we have to take antibiotics? And so most people ask, well, can't we just take probiotics to support recovery? But recent evidence suggests that probiotics taken after antibiotics may actually delay the return of the native microbiome. This included a lot of the butyrate producers, which was really notable. So essentially, they gave people lactobacillus and bifidobacterium-based probiotic after the antibiotics, and it delayed the return of the native microbiota, including the butyrate producers, for about 3 months. This was compared to the spontaneous recovery group, which recovered in 21 days, mostly. Obviously, there's still some degree of incomplete recovery, typically with antibiotics.
I just like to point
Yeah.
out that this is an example where like good evolutionary reasoning, like that would be a good, like that was a first guess. Like, okay, you can take antibiotics and be replenished with probiotics, but actually these real study, they're like follow up. Is that hunch? Would be really true, you know? I mean, but it's really, that's a good example of where, you know, you got to actually do the studies.
Yeah. Yeah. So if anyone's interested in that, they can read more in this blog article that I wrote about that study, or here's the link to the study in original form. It's possible, again, they use Lactobacillus and Bifidobacterium probiotics, so it's possible that Escularti or other probiotic formulations may be less detrimental or actually support recovery, but it's still unknown. So I'm quite hesitant about recommending probiotics after antibiotics now, especially since they're delaying the return of those butyrate producing microbes and probably stressing the gut epithelium more.
Which things like various fermented foods fall somewhere like under? Yeah,
that's a great question. I don't think we know. It probably could have a similar effect in delaying return if the microbes in the fermented foods are kind of filling up the niche so much that the butyrate producers can't return, then it's possible that could be detrimental as well. Yeah.
Is this just one study or is multiple?
This is just one study. It was shown at multiple levels. So they showed it. They did this in mice, humans, and they also then looked at what components of the probiotics in culture were inhibiting. And it was primarily the lactobacillus that seemed to be producing things that inhibited the growth of butyrate producing bacteria. So I'm hesitant to say, you know, this is definitive, you know, because like I said, it's possible that other probiotic formulations may have better effects. So yeah, but I do think, fortunately, we do have a better, more evidence-based approach, I believe, which is instead to take supplemental butyrate during and after antibiotics to support gut hypoxia. And so one of the studies that or going off of one of the studies that I mentioned earlier, taking butyrate during and after antibiotics might prevent that colonocyte energy starvation and attenuate dysbiosis. And in fact, they showed in that antibiotic study that I mentioned earlier that if you supplement the mice with tributyrin, which is basically a triglyceride form of butyrate that's targeted to the colon, or tends to be released more in the colon, it restored epithelial hypoxia in antibiotic-treated mice and reduced the fitness advantage of salmonella in the gut after antibiotics. So I'm really interested to kind of piggyback off this study. I really want to do one where we can give butyrate and see if it can prevent the entire effect of antibiotics on gut composition. You know, because this really only looked at salmonella and how that responded and looked at hypoxia. But if we look at the actual gut composition, can taking supplemental butyrate to support this pathway prevent the whole spectrum of antibiotic-induced dysbiosis?
You can take your rainforest analogy and do the same thing here and say, like, well, assuming if we, cut down everything, burn it off, well, what happens? Do we introduce one certain type of plant in which then now has free reign of the place?
Yeah.
Or do we want to put more nutrients in the soil? Or
I love that.
So, yeah.
Absolutely.
So this butyrate can be biased.
Yeah. So you can actually buy tributyrin. You can also, there's other formulations of butyrate that are more targeted to the colon. So probutyrate is one. I have no affiliation with them, but probutyrate is essentially in a fiber matrix, so it makes it to the colon and disperses there, whereas tributyrin is in a triglyceride type form. So both of those are commercially available, and you could take them during and after antibiotics. It's also possible that ketones, whether exogenous or maybe you just fast if you have to take antibiotics, and that could potentially support gut hypoxia by upregulating your ketone production. So I'm really interested in exploring some of these other avenues to be able to prevent antibiotic-induced dysbiosis. But perhaps even better would be stool banking and autologous FMT. So interestingly, this study actually had not just those two arms where they had probiotics or spontaneous recovery, but they also did a group where they did autologous FMT. So what they did is they had people before taking antibiotics do a stool sample and store it, and then the day after they finished the antibiotics, gave them a fecal transplant of their own stool to re-inoculate their gut. And they found that this autologous FMT restored the entire composition of the gut microbiome in less than a single day after antibiotics, even down to the mucosal level. So this study included looking at the lumen, looking at the mucosa, and biopsies. So this was really amazing and has incredible potential to prevent loss of diversity and the associated increased risk of chronic disease.
It's just, again, on the rainforest analogy, well, let's take a little sample of all the plants that are here now. Replant them where as soon as we burn it all down and they refresh.
I'm going to do that for my presentation next time. Thanks. Oh, it does. It definitely does.
It brought that one.
Yeah.
It definitely does. Yeah.
If you're taking antibiotics for some other, and there's the ones.
Right, but that actually brings me to my next point, which is how can we make this a standard of care to re-inoculate people with their own personal microbiome right after antibiotics and prevent increased risk of chronic disease. So this is actually a project Stephen and I have been working on a little bit for the last probably about 6 months. It's called My Gut Bank. It's a citizen science project and personalized stool bank. So the idea is that we have people submit their samples. We essentially store them at -80 degrees Celsius prepared for fecal transplant. And then if you have to take antibiotics, obviously personalized fecal transplant, not FDA approved currently, but the idea is that perhaps we could match individuals who want to store their stool samples and preserve their gut health into clinical trials to demonstrate the safety and efficacy of stool banking to prevent chronic disease.
When the FDA said that stool is a drug,
Mm-hmm.
they said the only time you can do fecal transplant is if you have 2 antibiotic treatments for C. diff.
Right.
So how are you going to get around that?
Well,
You're
that's
convince the FDA that they have to expand their indications for FMT.
Go to the Bahamas.
Or you
Yeah,
can go
I
to
mean,
England and get
I'm not saying it's easy, and that's why it would have to be done it would currently have to be done in a research setting. But you know, personalized FMT. Right now, the FDA does not distinguish between regular FMT and personalized FMT. But if you think about it, personalized FMT, you're only re-inoculating with stuff that you've seen before, right? So even if our screening techniques are missing things, you're not putting anything in that your body, that wasn't there before. So the hope is that perhaps, we can make that distinction. And I'm really interested in basically starting a discussion more about this, is because I think this needs to happen, to prevent chronic disease, especially you think about all the kids who are getting antibiotics at age 1, 2, 3, 4. My mom sent me my medical records the other day from age 0 to 4, and I had 6 antibiotics in that time. And that definitely did not help me with my, chronic eczema growing up. So I'm really interested in how we can leverage this to make this a standard of care, because in the future, there's really no reason why we shouldn't be able to say, okay, you're going in for scheduled surgery, right? We know you're going to get antibiotics. Why not bank your stool beforehand and give you a re-inoculation afterwards? So I'd love to hear you.